So now we will go to the last talk of the session that will be done by Marta Kapps, who is the leader of the Discovery Bioanalytical Laboratory at Hoffmann La Roche. And he will talk about the new approach for the microsample preparation. So I will leave the floor, if you can come, Marta.
Okay. Hello, everyone, and thank you, Pascal, for your introduction. So thank you also the organization committee to give me today the opportunity to speak about our new approach for bioanalytical microsample preparation with the Labcyte Echo. So I'm working at Hoffmann La Roche in the field of DMPK. So DMPK stands for drug metabolism and pharmacokinetics. In particular, pharmacokinetics describes how the body affects a drug after administration through different mechanisms of absorption, distribution, metabolism, and excretion. So I just see that the presentation, there are some problems with the format, but I'm apologizing for that. Sorry. So I'm working in bioanalytics, and bioanalytics is a discipline where the drug levels are quantified in biological samples. So a typical PK study, you have the in-life experimentation. So you apply a drug to a subject. It can be a mouse, it can be a rat, it can be a larger species. And then you sample blood over time. Those blood samples are then centrifuged, and the plasma samples are delivered to the bioanalytical lab, where I'm in charge of. And we are quantifying the drug in those plasma samples. So, and then at the end, with our results, the PK parameters are assessed. So why I'm here today is since few years now, our company, Roche, as many other pharma companies, are moving to microsampling. And why microsampling? Because you can obtain better PK results. So we can have now full profile versus composite profile. So in the past, we needed several animals, especially for mouses, to cover the full range of the time. So it's also less invasive, so the final results are less affected by the sampling technique. And the improvement of you improve animal welfare and easier animal handling. So no surgery is needed for cannulation, less stress during sampling, and this is also true for larger species. So you need you reduce the number of animals. This is particularly valuable for the transgenic mice, which are rare and really expensive. And you need also less drug to test your compound. So to understand the bioanalytical challenges, so I would like to introduce you to the technique we are using in our lab. So it's liquid chromatography coupled with mass spectrometry. So it's a so-called LCMS or LCMS-MS. So we have a pumping system. We have an autosampler where we inject our sample, and then pushing our sample through the column, so we do the chromatographic separation, and the mass spectrometer is there to get the signal from the drug. So a typical outcome of our LCMS system is a so-called chromatogram, where we have a plot from the MS response versus retention time, and the peak area of the chromatogram corresponds to is directly proportional to the concentration of the injected sample. What we have to know here is that MS response is also matrix dependent and compound dependent. So this means that for any analytical run, we need to calibrate our instrument. So for this, we prepare calibration samples in the same matrix as our study samples. So if we analyze mSample in a special strain, we use the same blank plasma to prepare our calibration samples. So a typical system looks like this, as you can see on the picture. So you have the pumping systems, you have the solvents on the top, you have the autosampler, where we put our plates with the samples. The column is really the smallest part, and this is the column oven. And then we have the mass spec in blue, where we get those. So once we have calibrated our instrument, so the study samples are analyzed against these are plotted against this calibration curve, and then we obtain our final concentration, which is used to assess the PK parameter. So, but prior any injection on our system, so we have to prepare our samples. So, as I said, we need calibration samples, but we need also quality control samples. The quality control samples are prepared in the similar way as our calibration sample, with the exception that they are used to to qualify our analytical run. So for this, we take 49 microliter of plasma. We add 1 microliter of DMSO stock solution containing the drug at different concentration levels. And once those samples are ready, then we do the final workup, which is a protein precipitation, because we cannot inject on our column proteins, otherwise this column would be blocked. So for this, we take exactly 5 microliter of plasma. We add 15 microliter of methanol or acetonitrile, the precipitation agent. We mix with centrifuge, and then in the pellet of the tube, you have the proteins precipitated, and we have the supernatant. And the supernatant, and we inject 1 to 10 microliter of supernatant into our LCMS system. In 90% of the cases or more, we need only 1 microliter. So if you see the volumes that we are using, 5 microliter plasma, it's liquid, 15 microliter of precipitation agent, so we end up with a volume, I would say a little bit less than 20 microliter of supernatant, and we need only 1 microliter. So here, this is very important, because here you see that you have still room of improvement if you want to miniaturize. But why are then our analytical challenges with the manual method? So we know that the quality of the results we deliver are directly linked to the quality of the pipetting step. So if you have variability on your pipetting step on the 5 microliter, you will find them on the results, on the final results. So that's why, by experience, so 5 microliter is the limit of what we that we can use manually in the lab to be in our quality criteria for our analytics. So if you have mouse studies, so the plasma volume we get, and this is the most of the studies we have, are with this volume, so it's between 5 to 10 microliter. So it can be 6, it can be 7, depending on the day. So there are days where it's easier to get the samples. And then you can imagine, then if an analytical run fails, you use 5 microliter, then the full experiment needs to be repeated. And this is really time-consuming and has also a big impact in research. So you can dilute also your sample. So this is a possibility, and that we use also, is if you have, for example, 4 microliter plasma, then you take 2 microliter, you add 8 microliter of blank plasma from the same matrix. You mix, you end up with 10 microliter that you can then you can use the 5 microliter, but you add an additional pipetting step, variability, and your limit of quantification is directly increased by the dilution factor. And this is particularly critical, especially for the PK studies, for the terminal points. And those points are the most important to assess the clearance of our compound. So some other challenges are evaporation. I will speak later about this. And the mixing is also a problem, sample homogeneity, especially if the plasma, you have low volumes, you freeze them. In the tube, when you freeze them, you have some droplets, water droplets, so you have evaporation that are fixed on the top of the tube. And you have to bring them down by centrifugation, mixing them. So it's really, handling low volumes are really critical. So what are our options? Do nothing, continue as how we are working now. So this is often frustrating, because if you have to repeat, you have to explain why. It's time consuming, because to take exactly 5 microliter, if you have only 6 in your tube, you can imagine that you have to pipette carefully. Dilute systematically, this is possible, but this applies only to high dose studies, tox studies. But as I said already, it has an impact on the variability of the result. Or the last way we wanted to go is to miniaturize the liquid handling process by assisting us with an automated system. As I told you just before, we have room of miniaturization to solve the problem there. So the system we evaluated here was a pipette-based system, Freedom Evo from 200 from Tecan. So we have many of them in some neighbors' labs. The volume range that you can handle here is 100 nanoliter. And it's a pipette-based system, so it's very similar to what we do manually in the lab, except that you can automate it, and it's probably more accurate. in routine. We tested also the inkjet-based system for HP D300 from Hewlett-Packard, which is a printer where you remove the ink cartridge, you replace it by a reservoir where you can put your sample, and then you can just print your samples into the plate. And the last system, it's the Labcyte Echo 550. The lowest volume that you can transfer there is 2.5 nanoliter. It's an ultrasonic probe going below the source plate. You apply an ultrasonic wave, and then you extract 2.5 nanoliter droplets, which are transferred to the destination plate top down. And yeah. So to understand the steps we wanted to automate it, here on this slide you see the details of the manual steps. So we have a first step, which is a serial dilution from the DMSO stock solution containing the drug. Once this is done, we spike 1 microliter into 49 microliter blank plasma in order to obtain our calibration and QC samples. Once those are ready, all the samples with the unknown samples from the study are prepared in the same way. So we do the protein precipitation by taking 5 microliter. Often we have a range between 5 to 50 depending on the volume we have. So it's clear that if you have a large volume, we use a larger volume to be more precise in our results. But speaking about microsampling, it's 5 microliter plasma and 15 microliter of methanol acetonitrile. We add also internal standard for the MS. After mixing centrifugation, so we put the plate in our system and we inject 1 to 10 microliter, as I already told before. So now we wanted to automate it, but we don't want to reproduce exactly what we do manually, because at the end we want to have a benefit. So for this, in a certain way, we do the dilution of the stock solution. And then we prepare directly the target volume of 1 microliter. So this was the first target to achieve, to prepare directly our calibration and QC at 1 microliter. And the unknown samples are then also, we transfer also 1 microliter, and we do the final protein precipitation with 30 microliter of methanol acetonitrile. So now just an evaluation summary and the reason why we have chosen the Echo 550. So with the pipette-based system, so we could cover the step 1, step 2, and step 3 can be covered, but we are coming in the same range of limitation we have manually. With the inkjet-based system, it works perfectly for everything what has to be done with the DMSO, but it cannot pipette plasma. So the most versatile system was the Labcyte Echo. So here we could cover the DMSO liquid handling as the plasma liquid handling. And we can cover all the 3 steps, and we were not at the limit of what we can achieve. So now I will focus more on the tests and optimization we did with the Echo. On this slide, you see the first test that we did. So it's a feasibility test. So we prepared 5 microliter calibration samples with the Echo, and we compared them with QCs prepared manually. And what we can see here is that you can see the result for 3 different compounds done in duplicate. In the lower part, so we had to discard some values, but this is due to the sensitivity of our LCMS method for those compounds. We have some outliers here. So those are the values you see are the accuracies. So for our quality criteria for LCMS analytic in discovery, so we have to be, the accuracy has to be between 100 plus minus 20%. And we can also discard 20% of our QCs and calibration. So with those results, so we are really in line of what we have to do. So we scaled down the method to 1 microliter. And here you see there were many other tests in between, but here you can see the results from 2 studies, 2 different compounds, analyzed twice on 2 different days. On the left side, you have the result, on the left side, you have the result for 1 compound. On the right side, the result for the other compound or the other study. So you have the 2 calibration curves from the 2 runs. For us, this is really perfect. You see the quality control, so they are within our quality criteria, no rejection for the different concentrations. You see also the result from our own samples, and here you see the difference between the two runs. So it's in between 5 to 10% of difference, which is acceptable and good for our purpose. The result from the second compound are really similar, it's just a mirror. So we wanted to know more about the transfer of these 2.5 nanoliter droplets. For this reason, we did an easy test, which is a qualitative transfer test. So we transferred an ink from the source plate, the ink droplet, a single droplet, to the destination plate, which was covered with a paper. And there you can see the spots of the ink on the foil. This test was repeated several times, and we observed always that the droplets were found where they have to be found. But we wanted to know more, so we are working in a field of quantitative analytics. So we did the same test, similar test, but to evaluate the quantitative transfer. So for this, we transferred 2.5 nanoliter orange G solution in the MSO. Orange is a colorant. And then we added 50 microliter methanol with the multi-drop. There is a similar instrument just on the exhibition. And you can see here the precision values that we have normalized the absorbance to 100, and you see then that corresponds to the precision. And in the first row, you see there are some outliers. In the first column, sorry. But this is due to the fact that the multi-drop starts with those as the first well, and probably the volume dispensed is a little bit lower than the volume dispensed in the neighbor wells. And that's why the values are a little bit higher. The real outlier we found are displayed here on this table in red. So for those, we don't have any explanation why it's like this, but at the end, it corresponds approximately to 0.5% of failure out of 384 transfers. So we decided just, according to those information, that we should, if possible, transfer always 10 droplets at minimum, so that in case one droplet fails, so that the error on our volume is below 10%, or equal or below 10%. So now we were ready to go live, and then for the studies where we had enough sample, we did a measurement with 50 microliter. This is where we are the most precise. And we compared it with the method of 1 microliter done with the Echo. This test was repeated several times, and we were really surprised to see that we had always an overestimation by 10 to 20% for the results obtained with the Echo. Well, it could have been also an underestimation by the manual method. But after some investigation, so we found out that the reason for this was a concentration effect due to evaporation. So how can we explain this? So if you have really low volumes in your well, so if evaporation occurs, this occurs on the top of the liquid. And on the top of the liquid, so the concentration of your drug will become higher. And it's also the place where we are taking our droplets. So the question is now, how can we solve this issue? So we found there are two options which are easy, that we could use easily. So the one is to cover the plate with a lid and to work faster, to prepare our plates. So in collaboration with our workshop at Roche, so they redesigned the plate, a lid with holes from 0.2 millimeter to 2.5 millimeter, knowing that the droplet of 2.5 nanoliter, if it's really spherical, the diameter would be 0.17 millimeter. So we are with the 0.2 millimeter, we are extremely close to the droplet size. So we fill the plate with water, we put the lid on it, we put it in the Labcyte. With the survey scan from the Echo, we measure the liquid high at the beginning of the experiment and after 25 minutes. And as expected, so depending on the hole size, so the evaporation is proportional to the hole diameter. And so we found a way to reduce the evaporation issue. But now the question is which diameter can be used so that that we still have a transfer from our droplets through the hole with this lid. So we did again the ink test. So we transfer the ink droplet. So on this picture, you see the results only from the plate, from the lid with the smallest holes. Because starting from 0.6 millimeter diameter and upwards, so we had a full transfer, no failure from our droplets. Starting from 0.4, so we observed 3 failures out of 96. And our surprise was really with the 0.2 millimeter. So if you know that 0.70 millimeter is a droplet size, we were still able to transfer 1/3 of the droplets. So this means that our workshop did an excellent job. And also the precision of the transfer from the Labcyte point of view is excellent. So to speed up the process, so if you see, this is the source plate that we have to fill manually. It's really funny to spot the right well where you have to put it's a 384 well plate. To spot the right position where you have to put your samples. So we imagined several options with the workshop, how we could do this and so on and so forth, but at the end the solution was extremely easy. And we found it on internet. And it's an application called Pipette Guide 384. For less than $10, you load it on an iPad, you put your plate, you select column, row, your wells are highlighted, and still you can change the color if you are not happy with the blue. But nevertheless, if you put a plate on an iPad, so the plate will slide on it. And this is also critical for 384. So we asked again our workshop, and they did a 3D printout that we could glue on the tablet so that when you put a plate, the plate will not slide on it. And I did also a tablet holder so that you are really with the inclination, so we are really in front of it, and you see our colleague who is pipetting, so you increase the comfort, and we are really fast, so we could really reduce dramatically the plate preparation. So one of our, what is also very important was the choice of the destination plate. So we don't want to transfer again the liquid from the plate which is coming out from the Labcyte to a new plate for the analytical plate. So we have approximately 30 microliter sample in our tube, so this corresponds to this level. And we want to take out the supernatant directly with our auto sampler. So for here, the conditions were we need wells as narrow as possible, if possible V-shaped, so that the pellet is less than 1 microliter. He stays here in the bottom of the tube and does not interfere so that we do not take it to inject it in our system. And the best compromise was found with this plate from Edgin. Now, as we like challenges, so we still scale down to 0.5 microliter because we saw that it's possible. And I think it's still possible to go deeper, so to go to 0.1 microliter. But there is no really need from our side, so that's why we stayed with this 0.5 microliter. And we did again a correlation between study samples measured with both techniques. So on the y-axis, you can see the concentration obtained with the Echo. On the x-axis, the result obtained manually. And for our purpose, this correlation is perfect. So you can still observe that the slope is 1.12. There we have still a certain, let's say, we have still 10% more as 1. So, and this can be explained simply to the evaporation effect of the sample. And this is really in a range which is really usable for our needs. With this, I come to the conclusion. So after some efforts, so we were able to scale down our method from 5 microliter to 0.5 microliter without compromise on the quality of the results. Sample preparation is now fully automated. And the leftover sample can be reused for additional measurement. So in case we have to repeat, we can repeat. We prepare also our plate in duplicate so that we can directly use them without any preparation in between in case we have to repeat analytics. And especially in our field, so in some compounds become important, so they want to do meta-ID, metabolite identification, and they can use the leftover sample to do their qualitative work without repeating the experimental part. And the amount of blank plasma needed to prepare the calibration and QC samples was reduced from 3 milliliter to 0.2 milliliter. This corresponds to 90% reduction, and can be directly translated to 90% less animals are needed to prepare this blank plasma. So I would also thank all the people from my lab, from Discovery Bioanalytics, and particularly Christophe Lamont, who did a lot of those tests, the people from the Roche workshop, as our contacts with Labcyte. So many thanks for your attention.
Thank you. Thank you, Martin. So is there any question in the audience?
Yes. Many thanks for your talk. Thanks a lot also to give us a good argument to ask for a personal iPad in the lab. I have a question about the evaporation effect. Have you tried to have or to control the condition under your robotic enclosure, for example, the temperature or the humidity degree? Is this something you are managing in your lab?
Inside of the instrument, for us, it's not possible because it's closed. There is water already inside, but it's cooled, it's tempered, so the temperature is constant. But outside, so we imagine also a way to put the plate to store them under saturated conditions. So those are options. But at the end, so we just try to work as rapidly as possible. So once the plates are ready, we just put it inside the instrument, we do our transfer. And this was the easiest solution. But I think it can be done, yeah.
Is there any other question? Well, I have a question, because you mentioned that with the evaporation, you have a problem that you could solve with the lid. I saw the problem was 10 to 20% of differences, and then at the end, in the conclusion, you accept 10%. So do you consider that you have solved that problem totally?
Well, it's not solved totally because you will always have evaporation. So we have to deliver results, I would say, with precision of plus minus 20%. So with we have a systematic error that we saw. If you would have, I would say, if you have good arguments that with microsamples you cannot achieve better, our customer would have accepted this also. But we were not really happy about this. So that's why we investigated a little bit more in order to reduce it to a reasonable level.
Maximum. Okay. Thank you.